All right, good afternoon, everyone, and welcome to our second episode of Vitamin C, your biweekly dose of marine education. Thank you for joining us. My name is Rachel, and I am the online marine education program facilitator for the Ocean Sciences Center this summer. And today I have Dr. Pat Gagnon joining me to talk about marine habitat mapping. By hosting these sessions every 2 weeks throughout the summer, we hope to develop a connection between the marine research happening throughout Newfoundland and Labrador and the public, and to hopefully encourage a continued interest in marine conservation, learning, and protection for the future. As I just mentioned, these sessions will occur bi-weekly all summer with numerous scientists so that we can learn about their experience with marine research and why this research is relevant and important to the quality and health of our oceans, both on a local and a global scale. So if you have any questions for Dr. Gagnon throughout this session, feel free to post them in the comments section and we will do our best to discuss them. And so again, thank you so much for joining us. And Dr. Gagnon, I'll let you get started.
Thank you, Rachel. So I'm going to start to share my screen. Welcome, everyone. Thanks for joining this presentation today. Let me just... You see my screen?
I can see it, yeah.
Perfect, thank you. Yeah, okay, so yeah, today I want to talk a bit about marine habitat mapping, which is one of the things we do in my research lab at Memorial. So that's a very interesting topic. It's also a kind of complicated one, so I'm going to try to keep everything as simple as possible. And as I go through my slides, I want to emphasize some of the tools that we use to map marine habitats. And I'm going to talk about scuba diving. I'm going to talk about remote sensing and a bit of everything that's kind of in between. So on my first slide here, what you see is basically a picture that's been taken with an helicopter and a standard camera, just a Nikon Coolpix camera. And I'm kind of using this image here to show you a few things here that are of interest for this presentation. So that's basically a picture of an island. So you see the forest here. That's the terrestrial side of it. And here you have the coastline and you see in the water different things. So it gets a bit pixelated right here. We have a few features right here and we have different colors around it. And as we get deeper into the ocean, we see that we have large patches of the same kind of brown material that we found in shallow water. And eventually we lose that color. So we get into something that's a bit more blue. Basically the idea is to use this kind of image to extract all kinds of biological information about what's on the seabed. So I'm going to try to cover the range of tools we use to extract different patterns from those imagery. So let's start by defining what's mapping. So very broadly speaking, if you look at the definition of mapping in a dictionary, you'll find that it's the act or process of making a map. So that's pretty simple definition. I guess we as scientists, researchers, we always like to make it a bit more complicated because we want to be very accurate at what we're doing. So we want to push it a bit further. So when you get into mapping, you use different kinds of information, which are kind of listed on this slide right here. So sometimes we'll use a combination of different things. We can use imagery from satellites, imagery from aircraft. We can send divers underwater to acquire some photos underwater. Sometimes we can use different devices which will use sound waves to basically map things on the seabed. And often when we go offshore, it starts to have like lots of sediment on the seabed. So we're going to maybe take some samples of sediment on board of boats with grabs and so on. So we like to combine all these sources of information to create some fancy maps of what's on the seabed. And ultimately what we're trying to do is come up with what we call digital maps. So basically maps that we can view on a computer screen. We can edit those images. We can analyze all kinds of patterns from this imagery. And ultimately we want to put that in the geographic information system known as GIS. So you guys are probably familiar with GIS concepts. I'm going to get back on that in a few minutes. But basically that's the ultimate outcome is we want to come up with a digitized map that we can play with in a GIS system. So who cares about those maps? Different kinds of people. Us first, as normal citizens, we always want to see what's on the seabed. We like to see what's there, but also policymakers, scientists, and researchers, they will use those maps to inform all kinds of decisions. So if they are after defining some regulation about managing a species, if they want to protect a different segment of the seabed in different areas, they will often use those maps, which are very useful again because they give us lots of information about what's on the seabed. So it really depends on the kind of instruments or device you want to use. And that will affect the quality of your map at the end. So what I'm showing on this slide here is you focus on the graph that's on the left. Let's imagine we're on the shoreline here, that's the 0 mark, and we progressively move towards the open ocean. On the x-axis here, it says we're covering a distance of about 800 kilometers from the shoreline. If you have a device that's, let's say, attached on a boat and you want to record the depth of the ocean as you move from shoreline to offshore, you'll get something like that. Let's say we have this profile that gives you the depth as a function of distance from onshore. And you see that with this kind of device here, you have very fine resolution. You see lots of details over very short distances. You have areas where it's deeper than other areas and so on. So you get the big picture here and it's very detailed. If you want to go the same distance, take the same trajectory with another device that's having like a coarser resolution. So taking samples, for example, at every 20 meters instead of every 10 meter, you may end up with a profile that's looking like this. So you're going along the same path, but you're creating different patterns. And that's just because of the scale of the instrument that you're using. So that's important to an ecologist because if you miss those features, if you take this, for example, here, when you use this device, it's very jagged. It gives you lots of details, whereas with this one here, it's kind of flat. So you're losing some details here, and sometimes you're completely missing some features like in this ellipse here, for example. So it's really important to select the right tool for the questions that you're addressing. So you might miss ecologically important fine scale features if you're not using the right tool. Now, once you get your, let's say, imagery of the seabed, it's one thing to know where the species are that you're interested in, but you want to also push it a bit further and try to explain why you find that species in certain locations and why it's not in other locations. So this kind of complicated graph here basically is there to show you the notion or illustrate the notion that basically you can measure different things in the environment that you know will affect the distribution of your organisms. In this case, they gave the example of could be the nature of the substrate. So is it soft? Is it hard? You could be interested in measuring temperature in the water, the slope of the seabed, the amount of chlorophyll, rugosity, and so on. So you could have these layers of information for the same location of the seabed. And ultimately, you take all these layers of information, you send that in a GIS, and you link those layers of information using some fancy advanced stats that are embedded usually within your GIS software. So making here, I guess, a complicated story simple, again, I'm coming back with the concept of GIS here. So basically a GIS is a software that allows you to create a spatial environment in which you can superimpose layers of information that help you explain the distribution of your organisms on the seabed. Now, with this said, I want to use a concrete example, something that's happening throughout the coast of Eastern Canada in rocky habitats is we find lots of those spiny soldiers here. So basically these guys are green sea urchins. And so if you dive in shallow water, let's say along the coast of Newfoundland, and you'd find that in Nova Scotia as well, in New Brunswick and Quebec, and it's pretty much the same seascape we find there, you'd see something like that. So in the first few meters of water, you see this band of vegetation here. So these guys are called kelp. So they're large brown seaweeds. They're very productive. They grow very fast. But at some point, if you keep going a bit further into the ocean, you'll kind of come across this line here where you start to have lots of urchins. So when you have this large, very compact aggregation of urchins, we call that an urchin front. Okay, so to the right of my screen here, you see kelp beds. So that's what you see at the top here. And right below it, you find urchin fronts. Now, if this keeps going forever, the urchins, they like kelp, and they will just keep going through the kelp, and they will eradicate all the kelp in that segment of the ocean. And what happens is that once this urchin frond has moved into the kelp bed, it leaves behind a different seascape, which we see here. That's called an urchin barren. So it's an area of the seabed where you could have had kelp before, but it's all gone because urchins have grazed all the kelp, and what's left is, pretty much only a few scattered urchins, which are not as abundant as in the front, but still abundant enough to keep all seaweeds from coming back in the system, except a few seaweeds. The one you see here is a beautiful brown alga called Desmarestia viridis, and this one can be there in the barrens because it produces some sulfuric acid that will repel the urchins, but it's just an exception here. So most of the other fleshy seaweeds won't be able to survive in the urchin barrens. So it's kind of a three type of community, if you will. So you have kelp, you have urchin front, and you have the barren in deeper water. Now, sometimes what happens in these systems is you'll have something that's going to come and kill the urchins. When this happens, the kelp will come back. They will be able to regrow and recover in the urchin barrens. So the urchin barrens will gradually disappear to the benefit of kelp. So kelp will just reintroduce the barrens. And that basically is a cycle. Okay, so we have a cycle alternation between kelp, urchin barrens, and this cycle varies regionally. So if you go in Nova Scotia, you'd have a certain cycle. If you come in Newfoundland, that's a different cycle, and there's different drivers for these differences in the rate of change between kelp beds and urchin barrens. I don't have time to go into that today, but just keep in mind that it's a cycle there. We have signs of ongoing changes though, because let's say, for example, in Nova Scotia, we used to have lots of kelp, and kelp are currently disappearing because there's another kind of seaweed that's coming in the system. So we know that with ocean warming, that's affecting kelp, which are usually cold water species. So just keep that in mind, is that we have this dynamic, but it's also changing as we move into a warmer ocean, I would say.
So, Dr. Gagnon, how would this cyclic pattern impact the ecosystem as a whole? Are there any ripple effects that can have an influence on other species as well?
Yeah, well, I mean, kelp are very important because they create a complex 3D environment that would not be there otherwise. And this environment is very good for other species of economical and ecological importance. For example, lobsters. Lobsters like to go in kelp to hide a bit. And there's other species that depend on kelp. And kelp also are good because they will incorporate the carbon dioxide from the atmosphere that gets into the ocean. So when they produce new tissue, they will take a part of this carbon dioxide and they will contribute to slow down the acidification of the ocean. So they have lots of different things that they do and that will help other species as they grow, for sure. Yeah, they're very important.
Perfect, thank you.
You're welcome. So now I'd like to basically get into some of the techniques and instruments we use for marine habitat mapping. So I'm going to start at the fine scale, which means that you're working on a very tiny area. And the benefit of doing that is that you can be very good at looking at what are the organisms that affect things over a very small scale, a fine scale. So I'm going to say a few things about scuba diving. I could spend a full lecture on that, but I'm going to limit myself to what we see on those pictures here. So what you see here is a diver that's kind of laying what we call the transect. So it's basically a line that we put on the bottom of the seabed, and we're going to measure different things along that line. So we use that because it helps us to be objective in the way we measure things. So instead of me going in the seabed and be attracted by something that looks really cool and I'm going to measure that, well, it's not the way we do science. So we want to put a line and just go along that line and be systematic at recording what we see on the seabed. What you see here to the right is basically a couple of divers which are deploying a sediment trap. So it's basically a tube that you put on the water. You let it sit there for weeks, months, and it will trap sediment in suspension in the water. Eventually, they will collect at the bottom of that tube, and all you need to do is bring this back in the lab and measure what's in it. And you can collect information about what's floating in the water column and so on. What you see here, lower left corner, is basically a little metal rod that we install on the seabed, and we use that to mark different things usually. and you see a blob of marine epoxy here. So marine epoxy is a kind of glue that will harden underwater. So we use those things as well to install different things on the seabed for habitat mapping purposes. And what you see here is basically a rack that we constructed ourselves just to hold vertical an instrument here that's measuring water currents in a 3D environment. So we get an accurate idea of what's the water flow in different kind of directions within the water column. So we combine these things together, and that helps us come up with fairly accurate maps of what's on the seabed. We often carry pretty heavy objects. What you see here is a couple of divers using a lift bag. So it's currently attached to a cinder block. And so again, just to show you the variety of things we do underwater, it's pretty dynamic. We use a range of instruments, but I'd say the most important thing when you're underwater is communication. So we have two divers here that are kind of solving an issue they have with a piece of equipment. So sometimes we carry with us a slate that we can write on underwater. So that's one way to communicate between divers. So there's all kinds of things we do underwater. And one of them, if we tie this back to the example of kelp urchin interface or dynamics, kelp barrens dynamics, what we see here again is that picture with kelp in shallow water, and we have a huge aggregation of urchins here. So what we like to do often is track the change in the position of the interface between the kelp and the barrens. And what we've done for this example here is we had divers measuring, we had a benchmark line here located somewhere in the barrens, and that was our point of reference. So a diver would go at different locations along that line and measure the distance with a measuring tape between that benchmark line and the position of the lower limit of the kelp bed right here. And by doing that at different intervals, you can come up with a rough map of where's the position of the transition between the kelp and the urchins. And if you do that at repeated intervals, so what we have here is a 2-year series here. We started in June 2000 and we finished in June 2002. If you take a look at the progression from month to month, you see that in certain times of year, the kelp bed is kind of receding or recessing, and in other parts of the year, it's actually recovering as we see here. So you can do that at different sites. It gives you those, again, rough maps, which still are good because it gives you very neat information about whether the kelp are doing well or not. And if you happen to do that at different sites, then you start to collect more and more information about a region, and that's how you move on with getting more accurate with your information. A few words about temperature here. So as you guys know, the ocean is sometimes showing some variation in temperature, and we like to record those variations, and we use different sensors for that. What you see here is a very basic one. It's called the hobo pendant. So it's pretty small, maybe 10 centimeter tall by maybe 5 centimeter wide, and we bring that underwater and we attach it to the seabed, and this guy will record water temperature for months and even years depending on the pace that you determine it's going to measure. So if you set it to measure temperature every minute, it will last long. The memory will fill up more rapidly. If you set it to record, let's say, once a month, then you can go for years. So it depends on the question you're addressing. So these guys, we have to install them on the seabed. So what you see here is a diver that's actually drilling underwater. So using a drill that you would normally not really use in the ocean, but that you can modify to connect to a dive tank. And so the air is coming from the dive tank and it helps the drill to work properly. You can drill some holes in the rocks, and then you put a metal rod and you attach your sensor to that metal rod. What you see here is another type of temperature logger. This one is more sturdy than the first one, and I tend to use this model when I'm in an area where there's lots of urchins, because urchins are very voracious and they will tend to even eat those plastic things. So this one is very strong, very tough, and I know that urchins will not graze on this one, but I've seen instances where this one had been damaged because urchins were munching on it. So it depends again on the type of application. I'm gonna go very quickly on this one just to show you guys that light that's coming from sun is interacting in all kinds of ways when it hits the surface of the ocean. So some of it is reflected back in the atmosphere, some of it gets in the water but get rapidly filtered out, and so that will ultimately affect the amount of light that gets to the seabed, and that in turn will affect the biology and ecology of a different species, including kelp. So I'm just mentioning because, again, keep in mind that um, things are different in the atmosphere compared to the bottom of the ocean in terms of light. There's lots of complex interaction happening there. So how do we go about measuring light on the seabed? Well, you can use the exact same sensor I was just talking about. So this sensor does two things. It records water temperature, but also light that hits the seabed. But the downside of it is that it just gives you a rough idea of the kind of light that you have on the seabed. It's just giving you an idea of the amount of light. It's not breaking it down in precise portions of light. The one at the bottom does it. So this guy I would bring with me underwater if I was to record light, to translate that into some biological aspects of the kelp. So kelps are photosynthetic organisms. So we want to know exactly what portion of the light they're using for their photosynthesis. So depending on the application, you're going to use different light sensors. Water motion is very important as well. So urchins, they respond to water motion a lot. If it's too wavy, they will tend to cling on the seabed and not move that much. If they do that, well, they're not busy munching on the kelp. So that's going to affect your dynamics here. So there's different, again, tools you can use to record water motion. I'm not gonna cover the details. I just want to insist on those two guys here, the one at the top here and the one at the bottom here. They're basically doing the same thing, but they're using sound waves to basically calculate water motion. So if these devices send a sound wave, eventually that sound wave will hit something in the water and will come back to the device. And it's the time it takes for the sound wave to leave the device, bounce on the object, and get back to the sensor that will determine the speed of the water in the ocean. So other kinds of sensors we can use are shown as well. That's a pressure sensor, which we can use to infer the water velocity. And this one is a homemade device, basically just a PVC pipe that you can get at any hardware store. You put some water in it and you leave some air in it, so it will just basically float vertically in the water column. And you can install at the top a little sensor that will basically measure the acceleration as the pipe is tilting with wave action. So all kinds of approaches we can use to quantify water motion. Sometimes we have to do stuff in the lab. It's not only about measuring what's in the ocean. Sometimes it's not really possible to do things in the ocean the way we want them. We want to do them. So what you see here is a wave tank that basically recreates the back and forth water motion in shallow coastal areas. So it's basically a 6-meter long channel in which we can put water. We have some concrete tiles in the center right here on which we will put our urchins, as you see in the bottom here. So we have urchins on the tiles, we have kelp here. So we basically created an artificial line of real kelp that we collected in the field, and we're kind of mimicking the back and forth motion of the kelp as it would in natural settings. And what we do here is we play with the wave intensity, and we look at how the urchins will tend to get together or deaggregate, and how quickly they will graze on the kelp depending on wave action. So that's one approach as well, is do that in the lab instead of doing it in the field.
So what would be a benefit to obtaining your fine scale measurements in an environment like a wave tank compared to doing so in the field?
Yeah, it's a very good question. So what we like to do in science and research is repeat our observations. We want to make sure that we generate what's called an average out of our observations. So we have a number that tells us more about the mean or average behavior of the urchins. If we were to do that just once, it would give us an answer for that particular observation. But maybe if you were to do it 5 or 10 minutes later, you wouldn't get the same response. So we do that in a wave tank because wave tank is ideal because it allows you to recreate the exact same wave environment over and over again. And so you can repeat your observations and generate very accurate, very reliable averages from multiple observations. There's no way you could do that in the field because the ocean is very erratic, and you can't possibly replicate your observations unless you stay there and you do it like right away. So that's why we do things in a wave tank often. When it gets to behavioral stuff, we like to control the wave environment very finely. So that's one benefit of doing things in a wave tank, in a controlled environment.
Perfect. Thank you.
You're welcome. So I want to just keep going with, gonna shift to intermediate scale. So the stuff I've said so far was fine scale. So looking at things at very narrow, over a very narrow window. So going intermediate here, we can use different kinds of camera systems. So that's a tow camera system that you would attach to a boat, and as you sail with the boat, you could have this guy go down in the water column, and there's a camera that's attached to it that's right here. You don't quite see it here, but it's attached somewhere here, and it's pointing towards the seabed. So as this guy floats in the water, it takes images of the seabed. You have diver held camera system. So that's a semi-professional camera that normally would not go in the water unless you put it in this special housing, which allow you to bring the whole thing underwater and keep your camera intact without flooding it and losing it. You have GoPros. You guys are probably all familiar with GoPros, so you can attach that on a stick or put it on your forehead and just, again, film what's on the seabed. So different cameras with different resolutions. I like this one here because it's heavy and it allows you for very good control. When it's heavy underwater, it's very stable, especially if you study kelp in wave environments. It allows you to keep your camera very stable. And that's my latest acquisition. It's a drop camera system, which I got manufactured on campus here at Mon. I'm gonna go outside of my slideshow for a few seconds here. Hopefully it's working well. So I'm just gonna move that back on my screen. And so another piece of research in my lab is we're trying to establish a foundation of green sea urchin roe industry for the province of Newfoundland. And we've used a camera system I was just talking about to basically map urchin stocks in the Bay Verde Peninsula region, which is up north in Newfoundland. So that's the map of northern Newfoundland here. So that's pretty much in this area that we've been assessing urchin stocks with the camera. And I want to show you a bit how the camera is working here. So that's the same picture as I have in my slideshow. So basically, that's the vessel we use for research in my lab. We have this camera here, which you see to the left. It's basically a tube in which you put your GoPro camera or Get Up camera. They're the same. So the camera goes in here. You insert the tube in that PVC plate right here, and the whole thing is attached to a metal frame, which in turn is attached to a cable here that goes into that reel here. And there's another component of the system, which is right here. That's a screen here. You have different controls. So you have someone that's sitting at the front of the boat who's watching the screen here, because we have the camera will stream live from the seabed. And what allows us to do is to basically lower down this whole cage in the water until it hits the seabed. And then we can get fairly accurate imagery of the seabed with this camera system. We have a GPS. So every time we take a picture, we take a GPS recording. And so that gives us an exact idea of where the picture was taken on the seabed. So just to show you quickly here how it's working. So we have someone on the boat who's lifting the cage. The camera is filming right now. It's getting in the water. And eventually it will drop slowly to the seabed. I don't know if you guys see that. There's a laser beam here. So we use that as a scale. So it's important to have a scale in your field of view. You need to know the distance within your image so you can retro calculate different kinds of things in your image. So as you watch, you see the camera going down to the seabed, and eventually it will reach the seabed. So that's a close-up view of how it looks on the seabed. So we see all the urchins here. Now we're lifting the camera because we're moving at the surface with the boat, going to the next location where we want to drop the camera on the seabed. And so we used to drop the camera every 20 or 30 meters as we were sailing on the surface with the boat. So that's what you get there. And every time the camera lands on the seabed, we will freeze the image back in the lab and extract all kinds of information from that imagery. And so what we did is we filmed the seabed over 12 kilometers, again in the Baie Verte area. And when we came back to the lab, we basically extracted images. And what I want to show you here is that we put all these images back on this map with the GIS. And if I zoom in here, you'll find pairs of points here. So if you click on this one here, you'll have access to the image exactly where the camera landed. Okay, and if you look at if you look back at the dot that's right next to it, it will open a little table. And in the table, you'll find all kinds of information. You'll find the person cover of the different critters that we found in this image. So we kind of processed all images this way, and we tabulated everything here so that if someone is interested in knowing how many urchins we found in that picture, or how many sea stars or mussels or whatever it was, it's all compiled here in this table for all of the images. And as I said, we covered 12 kilometers of coastline with that camera system in the boat. Did that at 4 different locations. And we use this information to determine if each of the 4 areas we were interested in had a potential, again, to produce enough urchins to extract roe and send that on the Asian markets. So that's another concrete example of things that we did using the drop camera system. Now I want to shift gear a bit. I'm coming to the end of my talk pretty soon, so I want to just say a few things about remote sensing, which is really allowing me to address questions at the broad scale. So we're talking about the kilometer scale here. So we're not talking about diving anymore. We're up in the air. We're using satellites or aircrafts. We have cameras attached to the belly of an aircraft, for example, which will take pictures along coastlines. And that's what you see right here. So on this image, we have a scale of about 50 meters. So every time you have this distance here, it's about 50 meters. And again, that's the land mass right here. That's along an island. And in the shallow water, you find those brown patches here. And for this project, what we did is we add imagery for 4 different years. So camera was flying for Parks Canada in that region here in 1983, 1988, 1999, and again in 2004. And we just thought we would take this temporal series and see what was the change over time in the extent of those black patches, which we suspected were kelp. And so we took those images, we put them into a GIS system. So these are the normal steps that we go through when we want to extract information about how things are changing over time in terms of kelp. Obviously, I don't have time to go through all those steps, but just to give you a broad idea of all the steps you have to go through. They're not complicated. Each of them is kind of easy, but there's a series of them. And so that's perhaps the downside of it, is that it allows you to map stuff over very broad scales, but there's lots of work behind that to get to that point. And I just want to emphasize here that even though you're using remote sensing, you still need to send divers in the water. That's important because these guys will do what's called ground truthing. So basically, they will take some images of the seabed, and we will take GPS coordinates of those pictures, and we will look at what they found compared to what the image taken with the aircraft is telling us. And if we have a match between what the diver think is kelp and what we see on the image, then we start to build a database that the software can use later to basically classify everything on the image that has the same color. So that's the idea here. So we can start simple with that. So let's say we have an image here. We have some land mass here. We have our black patches here, which we think are kelp. The first step would be to, again, move that in the GIS and apply the grid of points on top of your image here. And someone could go visually and classify each of those little points here as whether it's kelp or not kelp, or what they think is kelp and what's not kelp. Then you use this information. You do it for all of your years. So again, as I said, we had 4 years worth of data. And you can classify those dots as either kelp. In this case, it's red when there's kelp and it's blue when there's no kelp. And again, you can compare what's happening over time. And the percentages on top of each or at the bottom of each image here is telling us a proportion of all the seabed that was actually kelp. So everything that's red here. So in this case, it says 1983 was 37% of the seabed was kelp. In 1988 was 43, 1999 41, and there was a drop here between 1999 and 2004, so from 41 to 32%. So you can use this information and you can go a bit more fancy. So you can take those layers of information from the 4 years, put them on top of one another and come up with some fancy calculations that will tell you how stable things were on the seabed. So for example, in this map here, you have blue as permanent barrens, which means that uh, in the 4 years, it was always barrens, always urchins. Whereas when it's red, it's the other extreme of the scale. That tells us that it was kelp all the time in the 4 years. So that's start to be really interesting because you have a map here of the dynamics of how things were stable or not for this particular area of the coastline. So the whole point of this presentation is again to basically highlight the fact that scale matters. And again, we have a demonstration with this graph here. So it sounds a bit complex, but bear with me, it's simple in the end because all it does is that it gives us for each year, so 1983 to 2004, the proportion of the seabed that was kelp. So we see that for Ile du Havre, which was one of the islands that we studied, we had this kind of pattern here. So up, down, up, down. If you look at Ile Goéland was a bit different. So down, up, and then more stable and up again. So we see that there's a different dynamics for each island. But if you combine the whole information from all the islands and all the years, that's what you get here. So when you pull all the locations together, you find something that's very stable over time. So that's again, just to emphasize the fact that whether you look at things over the meter scale or the kilometer scale, you get a different message. So that's again emphasizing the fact that you really need to pay attention to the scale at which you're working when you do those things. Just a few words about getting even more fancy in remote sensing. So when I finished my PhD, I went into what's called the first research contract. It was a postdoctoral contract. For me, it was in the private sector, and the company that was hosting me had a very fancy camera system that you would attach to the belly of a Cessna. It's called the CASI 550. So this is a very advanced technology that will basically not only take one image as you would get with a digital camera, but it will decompose the image in as many as 256 layers of imagery for the same pixel. So basically what it allows you to do is get very, very detailed at what's on the seabed here. And we create all kinds of maps here using different algorithms, different sort of calculations with the GIS. And we would end up selecting the one that made the most sense to us based on our knowledge of the ecology of the different species that we were looking at. This map here is again was produced with the GIS. We set the surface all these kind of black lines here. That's basically the path that we took at the surface of the ocean with a vessel, which we equipped with a sonar because we were interested in also documenting the depth of the seabed. And so we use this layer of information about the depth of the ocean and combine that with the images taken with the CASI system to get even fancier and more accurate with our classifications, which is what we see at the bottom here. And so this is like generating lots of data, which in the end we use to produce what we call model of community dynamics. So basically, models that tells us like what's happening and when in the system and what kind of interactions we have between different species that we find on the seabed. This one is describing the dynamics for the urchins and kelp and a few other seaweeds. So it gives you an idea of how things vary over time, which is the axis you find at the top here in years, and the abundance of the different organisms on the seabed. So we have different processes happening here. So grazing by urchins, some competition for space between different species, facilitation, and we even know what's the impact of different parameters from the ocean, like water temperature, wave action, and ice scouring as well. So again, not getting to the details, but just to give you an idea of what's the ultimate benefit of taking all these measurements at different scales is that it allows us to draw a sketch of how things work and what species interact together, and what's the intensity of those interactions for the system that you're interested in. And I think with this, that's all I had time to say, I guess, as part of this talk. So I would like to thank you for your attention. And if there's any question, I'm happy to take them.
Perfect. Thank you, Dr. Gagnon. I don't see any audience questions coming in there just yet, but I do have a question or two for you. I wanted to ask you, what is your favorite instrument to use while on a scientific diving trip?
That's a good question. Like every instruments we use serve a specific purpose. And usually when we go underwater, we like to bring only what we need for that specific dive. So sometimes we bring camera systems. Sometimes if it's a dive for installing something on the seabed, for example, the drill, well, we'll just bring the drill and we'll make a few holes, install whatever we need to install. So I'd say I like pretty much all the instruments that we use because they serve a specific purpose. But I guess to make it very simple, and it ties back to the concept of diving, is that you have to be very very prudent underwater. You have to be very cautious, and there's all kinds of safety rules that that you need to observe. And for me, it's a very basic tool. It's basically my watch. So that's my Timex watch here. It's a very basic watch. And just to give you a little anecdote here, so I was for my PG, I was diving in one of the islands in the northern Gulf of St. Lawrence, and I lost my watch at the end of one dive. It was the end of July, and I had to get back to Quebec at Université Laval for my semester and stuff. And we came back in November for a few more dives. It was a break for us, so we dove there back at the island. I came across my watch just randomly, and that watch was still working perfectly fine. It was on time and everything. So I'm just mentioning because I think it's pretty cool that a watch like that is is good to go underwater for months, and it's very cold water. So I mean, this is very important to bring underwater because you have to respect your dive tables and so on. So that's just one, I guess, cool example here of how simple things are, but when they work well, it's perfect. So, but yeah, to get back to your question, pretty much all instruments I like to work with. Camera systems are cool. They allow you to take all kinds of information, imagery, and so on. Yeah, so I think that's basically it.
And I guess the moral of the story is make sure you have a high quality watch.
Yeah, exactly.
I did have one other question for you. I was just wondering if there were any major challenges that you've had to face during data collection, and if so, how you overcame those challenges.
Yeah, when you dive for kelp, it's usually in shallow water, and kelp tend to grow in areas where there's lots of wave action. So when you want to create the map I was talking about by sending divers measuring the distance from a benchmark line to the kelp line, it can be pretty exhausting. So you have to learn to work underwater. So what we end up doing for that particular application was to just wait for what's called slack tide. So don't go there when there's a tide. Just wait until the time the tide will shift, and that was a challenger. But that's how we ended up coping with it. Is we were only diving when we were at slack tide. So you got to learn. Got to get good at learning your environment and what are the changes that happen in the natural setting. So that's one challenge certainly for diving. Sometimes you dive in deep water. That's another type of thing I'm doing in my lab right now. Is we're studying rollits. If I have time here, I'm gonna show you a rollit. That's a kind of a seaweed that's growing on the seabed. It's very hard. Like I don't know if you guys will hear that, but it's very tough. It's got calcium carbonate in the cell walls of that seaweed. So we like to dive for those guys, but they tend to be found in deeper water. So you have to be safe on that side as well. So these are the challenges we deal with when we dive. So, but so long you're cautious and plan your dives properly, it's usually working pretty well.
Perfect. All right. Well, it doesn't look like we have any other questions coming in from the audience. I did have one question kind of for the audience. If I was just kind of curious if anybody has done any scientific or I guess recreational diving in the area as well. I know personally myself, I do have my dive certification, but I didn't get it here, so I've yet to experience the Newfoundland waters. But just out of curiosity, if anybody in the audience has done so. And do you have a particular place that maybe is your favorite to go diving around here, Dr. Gagnon?
We have sites in Bay Bowls. I like Bay Bowls. It's a shallow protected cove where we dive there and we find all the species that we're interested in. So that's probably one of my best sites. If we go for rollets, it's usually St. Philip's. So they are pretty cool sites to dive on. And just to add to your questions you had for the audience, if anybody is interested, we're teaching a scientific diving course here at MUN. So if anybody is a diver and wants to push it a bit further by learning all kinds of scientific skills on the water, we have this course that we teach usually in spring or summer, just so you're aware. Yeah.
Yeah, that is good to mention too, of course, in case anybody's interested in even further inquiring about that or registering for it themselves. But all right, well, I guess if nobody has any questions, then I guess that concludes our session for today. So thank you so much for joining us, Dr. Gagnon. It's been a pleasure, and thank you to the audience for attending. We hope you all learned something new. Stay tuned for some announcements in the coming week or so about our next session on July 21st. We'll be posting updates on our social media pages. So if you're interested in attending, just keep an eye out for those. And thanks again for tuning in, and we'll see you in 2 weeks. So have a great afternoon, everybody.
Thank you.